Today, I'm going to talk about post-translational modifications. What is translation? In molecular biology, translation is the formation of proteins from mRNA with the help of ribosomes present in the cytoplasm. This is in case of prokaryotes. Translation occurs in the membrane of rough endoplasmic reticulum in case of eukaryotes. The DNA produces mRNA which migrates into the cytoplasm for the synthesis of proteins. The proteins which are synthesized then folds or undergoes conformational change to perform their respective functions. Post-translational modifications, which is also called as PTMs. They increase the functional diversity of the proteome. This is done by covalent additions of functional groups of proteins, proteolytic cleavage of regulatory subunits, or degradation of entire proteins. These modifications include phosphorylation, glycosylation, ubiquitination, nitrosylation, methylation, acetylation, lipidation, and proteolysis. These modifications influence all aspects of normal cell biology and pathogenesis. Therefore, PTMs are very important in the study of cell biology and disease treatment as well as its prevention. Types of post-translational modifications. There are 4 types: trimming, covalent attachments, protein folding, and protein degradation. Trimming. Trimming is the removal of a part of a protein. Insulin is synthesized in the cell in its native form, that is, inactive. It cannot perform its function. For proper functioning, post-translational modification occurs that involves removal of a part of protein, which is called as trimming. This converts insulin into a 3-dimensional fully active form. The next type of post-translational modification is covalent attachments. Covalent attachments refers to addition of polypeptide chains. Proteins are modified by this covalent attachment for diversity of functions. And examples for covalent attachment is phosphorylation, glycosylation, sulfation, methylation, hydroxylation. First, let's see about phosphorylation. It is a biochemical process that involves the addition of phosphate group to an organic compound. Reversible protein phosphorylation generally occurs on serine, threonine, or tyrosine residues. This phosphorylation is thus very important for cell cycle, cell growth, apoptosis, and cell signal transduction pathways. The next type of covalent attachments is glycosylation. This has a significant effect on protein folding, conformation change, distribution, stability, and activity. The glycosylation is defined as addition of sugars that ranges from simple monosaccharides to complex oligosaccharides. The simple monosaccharide modifications of nuclear transcription factors and highly complex branched polysaccharide modifications of cell surface receptors. For example, oligosaccharides linked to asparagine, serine, threonine are major structural components of many cell surface and secreted proteins. This image shows different type of glycosylation. N-glycosylation. It is the attachment of an oligosaccharide, which is also called as glycan, to a nitrogen atom of a protein. O-glycosylation is the attachment of sugar to oxygen atom of serine or threonine residues in a protein. Glycation is the addition by covalent bonding of glycosyl phosphatidylinositol. This is found in surface of glycoproteins in eukaryotes as well as in some archaea. The glycosylation is carbon-to-carbon bonding to a carbohydrate molecule and a protein. Phosphoglycosylation is found in parasites is the linking of oligosaccharides to serine or threonine via phosphodiester bonds. The next type of covalent attachment is ubiquitination. Ubiquitin is an 8 kilodalton polypeptide consisting of 76 amino acids that is attached via C-terminal to N-terminal of a protein. After monoubiquitination event, there leads to a formation of ubiquitin polymer. This ubiquitinated protein is recognized by 26S proteasome that leads to degradation of ubiquitinated protein and recycling of ubiquitin for further post-translational modification of other proteins. Next type is methylation. It is the transfer of one carbon methyl group to nitrogen or oxygen to amino acids. This is mediated by methyl transferases and S-adenosyl methionine, which is also called as SAM. Methylation occurs so often that SAM has been suggested to be the most used substrate in enzymatic reactions after ATP. Additionally, N-methylation is irreversible and O-methylation is potentially reversible. This methylation is important in epigenetic regulation as histone methylation and demethylation influences the availability of DNA for transcription. The amino acid residues can bind to a single methyl group or multiple methyl groups to increase the effects of modification. The next type is N-acetylation. N-acetylation is the transfer of an acetyl group to nitrogen of a protein. It is N-terminal acetylation requires the cleavage of the N-terminal methionine by methionine aminopeptidase MAP before replacing the amino acid with an acetyl group from acetyl-CoA by N-acetyl transferase NAT enzymes. This type of acetylation is co-translational, that is, the N-terminus is acetylated on the growing polypeptide chain while it is still in contact with the ribosome. The 80 to 90% of eukaryotic proteins are acetylated in this manner. The exact biological significance is still unclear. Next type of post-translational modification is protein degradation. The proteins that are defective, for example, misfolded or destined for rapid turnover, are often marked for destruction by ubiquitination. We saw about ubiquitination process in the previous slides. The protein marked in this way are rapidly degraded by a cellular component known as proteasome, which is a macromolecular ATP-dependent proteolytic system located in the cytosol. The last type of post-translational modification is protein folding. Proteins must fold to assume their functional state. Folding can be spontaneous or facilitated by a special type of proteins known as chaperones. Finally, the detection of post-translational modification. There are several chemical and biological techniques for the detection of PTMs to find whether the modification has occurred or not. The methods involve mass spectroscopy, fluorescence staining of 2D gels, immune system activation by affinity binding DNA. That is all about post-translational modifications. Thank you.